Living things need energy to live and survive. For animals, they must eat plants or other animals to have energy. But how about the plants? How can they have energy when they can feed away animals too? Hi, I'm DJ. Let's find out the science behind this mystery. The sun is our primary source of energy. The green leaves of plants can use the energy of the sun to manufacture food. This food-making process is called photosynthesis, which means to put together by light. The leaves of the plant have the cells that carry out photosynthesis. If we zoom in on its cell, we can see its chloroplast, where the reactions of the photosynthesis occur. Photosynthesis consists of two primary steps: the light-dependent reactions and the light-independent reactions, or the Calvin cycle. The first step is the light-dependent reaction, which occurs within the thylakoid of the chloroplast. The membrane of the thylakoid disc have proteins embedded which carry out the light reaction. Outside is the stroma and inside is the thylakoid space. Light strikes the photosystem II, receiving photons or light energy. This light energy is transferred to a chlorophyll reaction center, causing electrons become energized and escape photosystem II, then move to a nearby electron acceptor molecule. To replace the electrons leaving photosystem II, water is split, releasing oxygen, 2 hydrogen ions, and 2 electrons. The first set of electrons continues to move down the electron transfer chain, releasing stored energy as it moves. This energy is used to create a hydrogen ion reagent. A protein in the electron transfer chain pumps hydrogen ions from the stroma into the thylakoid space, creating high concentration of ions in the thylakoid space relative to the low concentration of ions in the stroma. This gradient contains large amount of potential energy, which is used by an enzyme called ATP synthase. The hydrogen ions flow down the concentration gradient to a channel in the ATP synthase, releasing energy in the process. ATP synthase uses this energy to add a phosphate to ADP, forming ATP. As photosystem I absorbs additional light energy, the electrons again become energized, escaping photosystem I and moving down the electron transfer chain. At the end of this electron transfer chain, the energized electrons and hydrogen molecule are used to reduce NADP to NADPH. The light reaction released oxygen gas as a byproduct. The ATP and NADPH produced during the light reaction are used in the Calvin cycle. The Calvin cycle reaction occurs within the stroma of chloroplast using ATP and NADPH to convert the carbon dioxide to carbohydrate. The Calvin cycle can be divided into 3 phases: carbon fixation, reduction, and regeneration of RUBP. The first phase, carbon fixation, basically captures the CO2 from the atmosphere. CO2 is attached to RUBP, a 5-carbon molecule with the enzyme riboscope, resulting in an unstable 6-carbon molecule that quickly splits into 2 3-carbon molecules called 3-phosphoglycerate or 3-PG. The process of converting CO2 to carbohydrate begins. This phase is the reduction phase because we're adding electrons and energy to the CO2 molecule. During this phase, a sequence of reactions uses NADPH and some of the ATP from the light reactions. These molecules supply the needed electrons and energy for CO2 reduction. Electrons are added from NADPH, and through a series of reactions, 3-PG is reduced to form G3P, a carbohydrate. ADP and NADP+ return to thylakoids to be converted back to ATP and NADPH by the light reactions. One of the G3P molecules is set aside to be used as a building block for glucose, but the majority of the G3P molecules move forward to the third phase. In this phase, ATP is used to combine the rest of the G3P molecules to form RUBP molecules. This RUBP can then combine with the additional CO2 molecules, continuing the carbon reactions. The cycle has to turn 6 times to form glucose molecules. In general, plants produce their own food in the form of glucose through the process of photosynthesis and use this as energy to power its cellular activities and build new plant structures.